Thanks so much, Christina. It's wonderful to be here. As you said, I love open access events where everybody can register for free and we can get together. This has been the upside of this terrible pandemic, is we've realized that we can actually be all over the world and still be together in some sense. So this is, I love participating in events like this, and I'm happy to, put up my talk. I put it up on Twitter, and you can follow along the slides there. But you can also look at it just as I go along, and I'll show you the slides, but you can click through, and the advantage of having the slides is there are quite a lot of live links. So, and there we are. So this is the, these are the slides, and I'll just show a single page. There we are. There we go. And so I hope this is okay, and that you can see well. And so what I want to talk about is not too obscure. It's about something I really care about, which is interpretable methods for analyzing the microbiome. And the microbiome data are very complex. And from a statistician's viewpoint, there are actually really several interesting problems that come up. And we're often looking at dynamics of the microbiome. So we are in the search of transitions, but we try to do things with high resolution. And I'll show you an example of what I mean by that. But we want to try to detect the transitions just as they're about to happen. Now, we have models which in statistics are called hierarchical models, which have different levels. And we also have models where we can generate data by making a model of how we think things occur. Now, our quest is very often trying to understand the difference between something pathological and something healthy. And one of the difficulties that have come about when we analyze our data is it's not just one strain which is involved across many subjects. Different subjects have different strains of bacteria. And if you don't take that into account, you don't have very much power to detect differences. So let me go on to explain a little bit where the data come from. And this is funded by the NIH and by the Gates Foundation. And it's joint work with several wonderful students and postdocs I've had: Laura Simmel, Pratipa, Jenna Katan, Ben Callahan, Chris Sankaran, Julia Fukuyama, and the whole lab around David Relman. And we look at several things, and I'm going to talk to you mostly about pregnancy and the vaginal microbiome, but there are many different types of microbiome. Now, of course, as Christina said, I'm a statistician, so I come in with the view that we start off with the data, and then we estimate parameters from that data, and from that estimation procedure, we can go to some kind of generative model. And of course, I'm not going to explain all the details of this. It's laid out in a book that I wrote with Wolfgang Huber, which is called Modern Statistics for Modern Biology, which takes into account that standard parametric models like the normal distribution don't apply here. So you need something a little bit more refined. So what are the data? Well, in microbial ecology, we start off with reads, and so the features themselves, or the different species or ASVs, are not actually predefined. They're defined once we get the data. And every sample comes in with its own depth. And so we don't have an equal accuracy across the different samples. And then, of course, now we're integrating data about the immune system, about metabolomics, and many different layers in this picture of the ecology of what's going on. And we'd like to integrate that data and have an idea of our uncertainty in order to make conclusions and test hypotheses or give confidence regions. And so in the way in which we analyze the data, we do have wonderful tools already available, and the use of lists of different pieces of heterogeneous type can be put together either using phyloseq in Bioconductor or Multiassay experiment. And from a statistical viewpoint, we just see things as if they were mixtures, and that works really well. Now, in interpreting, we find that latent variables are really useful, and organizing factors or communities which are occurring where you have a co-occurrence of some of the bacteria. Now, one of the advantages of working with a reproducible research workflow is you don't have to stress about your choices. That is, you choose a method or a tuning parameter, but you can come back and change that choice because you have a script or a reproducible workflow which is adaptive. We do have to think carefully when you discard data because you won't be able to come back if you've discarded it. So the first problem or hurdle that we have is we have an infinite number of boxes or categories. And this work was done with Ben Callahan, where we realized that these reads, as they come in, they're never exactly perfect. There's some errors which occur. And we have to do a certain amount of denoising in order to get the most out of the data and keep it at the highest resolution. So we'd love to have strain level information instead of agglomerating data into OTUs. So this is a little bit like words that you have and Google guesses from your typos and the frequency with which those words occur what you mean. So here, for instance, you start off, you get a lot of these purple ones, and there's some of these other ones which have a few errors in them, but they're really very close to the purple one. So we're able to assign them to that bin. And so this correction, this error correction, which occurs, is easy to do if you don't forget about the frequencies. And so here, these are the bins, and this would be ASV1, which is the most prevalent. And then we have the other ASVs which occur. Now, before, people used something called OTUs, and they were creating similarities, say 97% similarity, and you make a circle and say, okay, this is OTU2 and this is OTU1. But the problem there is this procedure, this way of making a fixed radius, doesn't take into account that here, for instance, the blue are 100 times more frequent. So if you have, on one hand, 100,000 sequences of this type and 1,000 in this, you're going to have many, many more opportunities for error. So the variance of your whole cloud was much, much bigger. So having a fixed radius of 97% doesn't make any sense. So we use a little probabilistic model, and we go, the data we actually read, we denoise it, and this is what data 2 does, so we can go back to what the original sample sequences were. And I'm not going to belabor how that works, but it's behind a tool which has become very, very popular. And the data 2 package is very popular in Bioconductor, this error model. And for the problem at hand, looking at the vaginal microbiome, one of the most important species is Lactobacillus crispatus. And we see that if you use a standard method, you only see one OTU, one taxa. But if you look at this high resolution method, we're able to see many different strains. And each of these blocks correspond to the same patient. And so we can see there's a small change in the different strains for the patient, but mostly the patients keep the same strains. And you do have a lot of strain sharing across different patients. And we'll see that this is really important. So at the end, we have an ASV table with these new species variants or Amplicon strain variants, and a certain number of samples in which we count the reads. Now, what's important about this is these weren't predefined. These came from the data. And very often we have many more features than we actually do samples. And so you have to use nonparametric methods. Now, you don't summarize this data by relative abundance because you'd lose a lot of information about how variable or what the number in each of the number of reads in each of the samples are. And it's also very important to take into account what the read signatures are when you're looking for contaminants. And we use methods which use the actual counts. We don't have to look at relative abundance. But most of all, I think it's very important to remember that even though the sum of the proportions sum to 1, And so the parameters that you estimate, in some sense, they're all proportions and they sum to 1, so they are compositional. The data themselves are just numbers of reads, and the data are not compositional. And so that's something important to remember, because it has a big effect when you go to do the testing. Now, for the practicalities, what do we want to try and do? Well, we have heterogeneous data types, and so we're going to put them all together, and we have this data2, and we're going to combine that with the phylogenetic tree and all the data about the samples, and we put this together into a package which is called PhyloSeq, which allows you to do statistics on the microbiome data. And so you could do longitudinal data analysis, spatial data analysis. You could do all the different methods that exist, for instance, for ecology on the microbiome. And that's the big power, I think, of PhyloSeq, is that it uses tens of thousands of packages which already are available to us within R for doing statistics and even clinical statistics on the microbiome. And so we can also visualize the data. And I always say it's really important to stay in touch with your data and not lose it in a black box. And so plotting a heat map or a network of interactions is a very useful way of understanding what's going on. Now, in our new paradigm, when we're trying to integrate more than just the microbiome, we also have measurements of cytokines or metabolites, the metagenomics. So we have many different elements. And in particular, if you're trying to understand the vaginal ecosystem in pregnant women, you have many sources of information, as well as what we call the sample data is really essential. It's the clinical history of the women, and that, knowing whether a patient had already had a pregnancy or not are all important factors into understanding the vaginal microsystem. So you see that you have what we call multi-domain data of many different types. And there are these wonderful tools which allow you now, they're called Multiassay experiment, and they're containers in Bioconductor, which we use for doing all the analytics. Now, I'm just going to show you a little bit of an example, and this is the study of longitudinal states in the vaginal microbiome. And in this study, we had 49 pregnant women, 40 of which we take as our main case, and then the 9 other cases are our validation set. And what is well known already for the vaginal microbiome is that you have different state types. So here we have 5 state types, and I'll just point out to you that all 4, the 4 ones except state type 4, so 1, 2, 3, and 5, they're dominated by lactobacillus species. So here you have crispatus in group 1, and we have iners in group 3, and gasseri in group. So you have these lactobacilli dominated groups. And then here we have something which is very diverse. We don't have one dominant species. And this is a case which is interesting, because it's a case where we have diversity is pathological. Usually in the microbiome, diversity is associated to a healthy environment. In the case of pregnancy and the vaginal microbiome, it's the other way around. This huge diversity is the place where many of the preterm births are occurring. So you see these pink lines here correspond to samples which come from women who've had their babies early, and you see they're associated with this high diversity. And in fact, this community state type is not a cluster by any meaningful definition. It's just the absence of similarity. That is this huge diversity. And it's in fact the understanding of this group 4 which led us to these topic models. And I will just show you, these are the blocks of similarity of the community state type. And you just see that in state type 4, you just don't have a very high similarity. So we wanted to ask, could we predict the changes, in particular in and out of this state 4, which is more associated with preterm birth? And so if you go back to here, we don't see a very clear signature, but we did take out and look at just state 4 on its own and look at the strains which were associated with preterm birth. And we were able to see, and you see this association here, that state 4, they're much shorter pregnancies, that we found that the Gardnerella abundances were high in this community state 4. And so we can go back and try to understand much more about this lack of clustering and this very diverse state, but we needed new tools. And the new tools that we're going to show you in here, these are the 2 strains of Gardnerella in a replication study that we did, which showed that this study had shown, really stands up to different studies. Different groups of women give the same results. We have the very, very similar layout of the different strains. And we would like to interpret what's going on. So the challenge of this very complicated data is we have interpretability. Now, if you want a very simple, what we call balls and boxes model, then you'd have to say that the species are, they could be put into these boxes and all the boxes are independent. Now, that's not true. That is what we see is some taxa are quasi-exclusive. If you have Lactobacillus crispatus, you have very low chance of having Gardnerella. Or you have syntrophy, so you have co-occurrence. And the co-occurrence means that you have high probability of having one bacteria if another way is present. It's often because they need each other. So an ordinary multinomial model, what statisticians call the multinomial model, where the boxes are independent, doesn't work. And so we use a mixed membership model, which was something which was actually inspired by what people use when they're analyzing text. And we use natural language processing. This is actually something also used in genetics, but we use the parallel between document topics and community analysis. And this was work by Chris Sankaran in his PhD thesis. They make the analogy between, so we have the reads, and from the reads, that's the equivalent to words. And then we can go to the ASVs or the species. And that's the way that you go from words to terms. And as you see the assembly of these terms into topics where you have co-occurring words, which happen a lot, then it's the same thing. That is with the bacteria, they're co-occurring in communities. And as soon as you have this analogy, you can understand that in some sentences, you use different words for the same meaning. And that's the same. That is, some bacteria play similar roles, but they're not the same ones. And your sentence has the same functional meaning. And so this strain switching which occurs sometimes defeated standard differential abundance methods. But by taking the topics, we have much more flexibility and robustness to understand. So we have a statistical model, and of course this is a very short talk, so I won't go into all the details of the statistical model, but you can look it up. It's called a latent Dirichlet allocation model. And these were actually used for mixed membership in genetic populations by Jonathan Pritchard and co-authors Matthew Stevens and Peter Donnelly in the year 2000. So it's already been used effectively in biology. It's just many people have rediscovered it. David Bly rediscovered it for language, and we found it a very good model for the microbiome. So here is the multinomial that you have, but has hyperparameters. So we're changing things. So we're able to have co-occurrence, and we don't have these independent different boxes. Now, differential topic analysis does help with the strain switching problem. So if you have some strains which are synonymous of others, you can change your testing question. Instead of saying, do these two groups have different ASVs, we can say, do these two groups, the healthy and the preterm groups, for instance, have different communities? And see how the differential topics occur instead of just individual strains. Now, this requires us, in fact, to align or register the topic. And this brings me to the work that we did to deepen our resolution of the vaginal microbiome. This was Laura Simul who led this, and this is the multi-domain data assembly analysis. And so this is a paper that just went up on the archive and that's just been submitted. And in this paper, she discovers or associates subcommunities of the vaginal ecosystem, both in pregnant and non-pregnant women. And I'll just show you a picture of how she aligns the topics. And so if you only have one topic, you have all the species together. If you have two topics, you're going to split off into two different sets of topics. And you have different sets of bacteria. And then as you multiply the number of topics, you can go and you have groupings of the topics of bacteria which are co-occurring a lot. Think of words which co-occur. So for instance, words which have a lot to do with sports like ball, park, bat, and so on, they're going to co-occur a lot in, say, web pages which have to do with baseball. And so we have the same phenomena, we can detect them, but this alignment method allows us to see which of the topics which actually are stable across many analyses. And so this tool, which is a beautiful tool, which was done by Laura and Julia Fukuyama and Chris Sankaran, allows you to see that. And so here, for the case we're interested in, we have a succession of topics at varying resolutions, and we can also compare, for instance, between pregnant and non-pregnant subjects. And so we have this lactobacillus inus topic, which occurs in non-pregnant, and it occurs in pregnant, and we have this blue topic. Which is the same. And then we have some of these troublesome topics which involve Gardnerella. They occur both in non-pregnant subjects and in pregnant subjects. So you see these co-occurrences, and we also have these synonymous, the strains which replace each other, which are present in the topics. So this is a very useful way of seeing that. And we can also do the strain switching and do the differential topic analysis. Now, of course, I don't have all the time to show you all the different tools, but one of the things I wanted to say, and Christina mentioned it when she introduced me, is we really care about improving our reproducibility. And when we did this analysis with the topic analysis of the microbiome, vaginal microbiome, it's like an iceberg. That is, what you don't see is all the possible choices of methods that you could have chosen. And so, for instance, there are about 200 million ways in which we can analyze the data. Different normalizations, how you take the dimension reduction, all the different choices you have to do. And I have a talk where I show in the enterotype data, for instance, all the different ways that were possible, and it makes the data analysis very hard to reproduce. Now, in order to make it transparent, what we have to do then is to show all our work. And underneath of the iceberg, what's hidden is, I showed you the paper we just submitted. In that paper, there's a 227-page supplement, which was generated with all our R scripts and visualizations, and all the data analysis is available, and you will have access to the code, so you could try it out on your own data and decide whether or not, you'd prefer one method or another. But that's the way that we go around the problem of too many different analyses. So I put in my slides links to the reproducible research supplements, and so that you can look at some of them yourself. And so for all the studies we do, we have all the code and all the data, so you can try them out. And that's also true for the book with Wolfgang, which is open source. And of course, we have a whole series of packages and resources, PhyloSeq and Dada2. The tool that I just showed you, which does this beautiful alignment of the topics, is called Alto. And that's also available now as an R package. And then we have methods for decontamination when you have small amounts of DNA, and methods for integrating trees. And I haven't talked about many of these tools, but they're still all available. So what did we learn while we were doing these analysis is it's very important to understand the raw data so that you make an appropriate model and you make your approximations in a conscious way. That is, you have to make approximations, but you have to be responsible for them. There's not one way of analyzing microbiome data. The vaginal microbiome is very, very different from the gut. The number of taxa involved is very different. The library sizes are very different. You have to tailor your library and sample sizes to the problem at hand. The choices are not forever. As I said, you can go through and redo many analysis to see for the, using sufficient statistics and showing your code. What I showed you was an example of topic modeling. That's what we call in statistics a hierarchical model, in which you have latent structures which are easy to understand. But when you do that repeatedly, you have to have a method for registration and alignment. So your analysis is only as good as the explanation, the software that goes with it. And that's why it takes my group, very long time to publish papers. And I'm very thankful for the people who have the patience to work with me. And so here's our group with David Relman and many of the people who are involved in the studies that I just showed you. And I've benefited enormously from the tools and schools of statisticians who came before me. And I'm very thankful to the people who developed R, STAN, Bioconductor, and the community around that, and in particular Wolfgang and Rob Gentleman, who've been instrumental, of course.